Good morning, everyone. Myself Mantasha Hassan, and today we are going to discuss about the biomembranes or the cell membrane. Now, what is cell membrane? Cell membrane is the outermost covering which is present in both plant cell as well as in animal cells. Now, cell membrane is composed of many things like lipids, proteins, and carbohydrates. But basically, as cell membrane is made up of proteins and lipids, and we have 52% proteins and 40% lipids. These lipids could be phospholipid, cholesterol, or glycolipids. Phospholipid means the lipid which is attached to the phosphorus, cholesterol, and we have glycolipids. So it is a universal structure, and all the membranes of prokaryotes is similar to the eukaryotes. So cell membrane, it is also known as biomembrane, or it is also known as selectively permeable membrane. We can say it is a selectively permeable membrane. Now, what is the structure of biomembranes? We have sandwich or trilamellar model, which was firstly given by Davison and Daniel. And according to this model, plasma membrane is made up of three layers. Outermost layer is a biomolecule of phospholipids which is sandwiched between the two single layers of proteins. And according to this model, this protein layer is 20 angstrom and a thick layer of phospholipid is 35 angstrom thick. So this method, it is also called as amphipathic molecule because of the two types of pass, phospholipid, and we have hydrophobic philic tail and hydrophobic tail. So this is the model which was given by Davison and Daniel, and according to it, this plasma membrane is made up of three layers, in which there is a layer of lipid which is sandwiched between the two single layer of proteins. So we have two single layer of proteins on the outside, and we have two layers of lipids which is phospholipids, and the thickness is 75 angstrom. So that is why this biomembrane, this phospholipid molecule is also known as amphipathic molecule. Why? Amphi means both. So we have two types of parts. We have hydrophilic head and we have hydrophobic tail. Next, we'll talk about unit membrane model, which was given by Robertson in 1959. And according to this model, the cellular and organelle membranes are structurally and functionally similar, difference in chemically and size. So the detailed structure was experimented by electron microscope after the discovery. So we have fluid mosaic model, which is the most accepted model, and it was given by Sinclair and Nicholson in 1972. It is also mentioned in your NCERT textbook. So what do you mean by this? That protein icebergs in a sea of phospholipid, or it is also known as gulab jamun protein in a concentrated solution phospholipid of sugar. For example, we have a sugar and we have phospholipids, and what we have in that, we have proteins in that. So phospholipid is a main component of cell membrane, and it forms continuous structural frame of cell membrane. So we have such phospholipids. Next, the studies show that cell membrane is composed of lipids, and the lipids are arranged in a bilayer. Also, they are arranged within the membrane with the polar head towards the outer side and the hydrophobic tail towards the inner part. So this ensures that the non-polar tail of saturated and unsaturated carbons is protected from the aqueous environment. So phospholipid मेन कंपोनेंट है. phospholipid layer fluidity देता है to the plasma membrane because phospholipids are rich in unsaturated fatty acids which are lipid in nature. So what is the actual structure? That is quasi-fluid nature of lipids. Now the fluid nature of the membrane is important in cell growth, formation of intercellular junction, endocytosis, that is phagocytosis and pinocytosis. Then we have secretion, cell division, etc. Now cholesterol is present in the plasma membrane, and cholesterol are more rigid than phospholipid. So it helps in the stability of membrane structure. Cholesterol is absent in the membrane of prokaryote, and we have hopenoids which are present in the prokaryotic cell membrane. We have two types of proteins. Two types of proteins are present in the plasma membrane. First one is integral, which is present inside the layer of phospholipid, that is intrinsic proteins. So they again are of two major types, partially buried and totally buried. So some integral proteins which are totally buried, they are known as tunnel or channel proteins, and second we have peripheral or extrinsic proteins. They're superficially arranged on the surface of lipid layer and can be separated easily. So when they are buried completely throughout the layer, so they are two types of protein. So this is the overall structure of your fluid mosaic model of plasma membrane. Then we have active transport method, and then we have passive transport method. Thank you everyone.